Hi everybody, this is a video about action potentials, the setup of neurons, and a couple other fun things like that. You'll see a drawing that I made on the board of a neuron, actually a series of neurons, just a couple different things that we can review. Remember that when we're talking about neurons, these are going to be the dendrites, and this would actually be an example of a multipolar neuron. Multipolar means that there's going to be multiple dendrites and a single axon going down. The axon is this portion. This is actually going to be a myelinated neuron. You can see that there's myelin sheaths. In between the myelin sheaths is the nodes of Ranvier. We have the synaptic bulb, or the synaptic terminal, or the synaptic bouton at the end. This would be a synapse. There's also a synapse over here between the axon of one neuron and the dendrite of the next neuron. It doesn't have to be a neuron, so this could be a muscle, it could be something else, but generally it is going to be a neuron on the other side, especially if we're talking about interneurons. Remember that interneurons are going to be the go-between, that are going to help with processing and shifting things from one place to another. We do have the nucleus in the middle of the soma, the cell body. But if we're talking about how neurons actually function, generally the way that this is going to be broken down is in 4 easy steps. Step 1, we have the reception of a neurotransmitter from ligand-gated channels or chemically gated channels, the receptors. In a dendrite, in this example, we have sodium going through a chemically gated channel, so the CG. That would be some sort of receptor that would be permeable to sodium. So an example of that would be the nicotinic acetylcholine receptor, or any other sodium channel, ligand-gated sodium channel. So we have our neurotransmitter that's released into the cleft that's going to bind to the chemically gated channels or the ligand-gated channels. Sodium is going to go in following its electrochemical gradient. That is going to cause a positive charge inside of the cell. If that positive charge is big enough to reach threshold is the voltage at which voltage-gated sodium channels are going to open. Sodium is going to rush in. That's going to open up the next voltage-gated sodium channel. Sodium's going to go in. It's going to use myelin sheaths as a form of insulation to kickstart the charge and force it to go further down, further away than it would normally, opening up the next sodium channel, which is going to open up the next sodium channel, etc., and so forth, until you get to the synaptic bulb. At the synaptic bulb, a voltage-gated calcium channel is going to open. Calcium will go into the cell. That calcium will then bind to synaptic tethering proteins. Once we hit those synaptic tethering proteins are going to shrink, and the vesicle is going to merge with the membrane, releasing the synaptic contents or synaptic vesicle contents into the synapse. Generally, this is going to be a neurotransmitter that is going to interact with the next neuron or the muscle. In the case of a muscle, it's always going to be acetylcholine, and it will always bind to the acetylcholine receptor or the nicotinic acetylcholine receptor on the muscle cell. To rewind a little, if we go back to the axon hillock, and when voltage reaches a specific amount at the axon hillock, those sodium channels are going to open, and then we induce what's called an action potential. An action potential is an all or nothing type of response. It either occurs or it doesn't. Once we hit the threshold of negative 55 millivolts, the voltage-gated sodium channels that we talked about are going to open. Then, after a little while, once it hits the peak of the voltage-gated sodium channels, voltage-gated potassium channels will open. They're slower, and they initiate the repolarization phase. I've tried to color code things in here. Once it gets back down to resting membrane potential, then it goes into the end of repolarization because too much potassium has left, and it hits hyperpolarization. Finally, the potassium channels are going to close, and that allows the sodium-potassium ATPase, or the sodium-potassium pump, to kick back on and move the sodium channels out, and we return to resting membrane potential. We can look at this in a slightly different type of orientation, and you can see what's going on at the channel level. At resting membrane potential, there's more sodium on the outside, more potassium on the inside. When we hit depolarization, that initial portion, or once we hit threshold, sodium channels open and sodium goes into the cell. After that, the sodium channels will close once we hit the peak. The potassium channels will open and potassium will leave the cell. And they do a little bit too good of hyperpolarization and then returning back to resting membrane potential.